Hi, this is Miss Hammonds for AP Biology 2113. This is Chapter 2, Part B. It's a continuation of the chapter that we discussed in the last lecture note. So make sure that you have your guided notes available to support your understanding of this presentation. So we'll begin by talking about biochemistry. This is the study of the chemical composition and reactions of living matter. Organic compounds are always going to contain carbon. Carbon is always covalently bonded to whatever organic substance it's present in. Some examples of inorganic compounds is that you would have salts, water, acids, and bases. Some examples of organic compounds would be your carbohydrates, fats, proteins, and nucleic acids. Both inorganic and organic compounds are essential for life. And we're going to briefly look at some of the characteristics of water. Do know that water is the most abundant compound. Water has a specific heat capacity. It has a high heat vaporization. It's reactive. It goes through both a hydrolysis process and a dehydration synthesis. And it provides cushions around organs, specifically found in the cerebrospinal fluid, or CSF, surrounding the brain and the spinal cord. Water makes up about 60 to 80% of the volume of all cells. To speak more in detail about the characteristics on the screen, when we said high heat capacity, water absorbs and releases large amounts of heat before changing temperature or transforming its state of matter. It has a high heat vaporization. As water evaporates from liquid to gas, it takes the heat with it. As we mentioned with the reactivity, it's an important reactant in chemical reactions. And remember, the reactants are the side that is combining together chemically, and the products is what is the result of that reaction. Hydrolysis is when bonds are broken and water is added. Dehydration synthesis is when bonds are formed, water is then removed from that compound. And as we spoke earlier about cushioning around organs, specifically the cerebrospinal fluid that surrounds the brain and the spinal cord. All right, water is a polar solvent. Water is polar and will attract and surround solute particles. It causes ionic compounds, acids, and bases to dissociate. And when we say dissociate, they will begin to dissolve and become smaller particles. Salts are substances that, when dissolved in water, again dissociates into ions other than hydroxyl, or forgot what the OH one stands for. But what type of bond is found between sodium and chlorine? So in our last presentation, we talked about ionic bonds, where the electron is either going to be given away or it's going to be received. So the bond between sodium and chlorine is going to be an ionic bond. What type of bond is found in water? That's going to be a covalent bond, because the hydrogen atoms are sharing electrons with the oxygen atom. How does water with molecules interact with salts and why? So acids and bases. An acid is a substance that releases hydrogen ions in a solution. And bases are those, hydroxyl, I couldn't think of that. Bases are those substances that release a hydroxyl ion in a solution. One that accepts hydrogen ions already present in the solution. And with acids and bases, how they react with different substances depends on the acidity or the basicness of that particular substance. So here's our pH scale. pH is a measure of the relative acidity or alkalinity of a solution. Pure water has a pH of 7. And then on this scale, something that is closer to 0 has a high acidity, categorized in that range as battery acid, hydrochloric acid. Lemon has an acidity of 2 along with vinegar. Grapefruit juice, soda, tomato juice has a pH between 2.5 and 3.5. So from black coffee towards the battery acid, all of these items are acidic. The lower the pH number, the more acidic the object is. Blood has a pH of 7.4. We mentioned that water has a pH of 7. So seawater has a pH of 8, baking soda 9.5, bleach and oven cleaner is 13.5. So as the substance pH increases, it has a more basic range on the pH. Here are some examples, again, of pH. Please make sure that you are familiar with the items that are listed here. You may be asked to identify whether a particular object that is present will be classified as basic or acidic. Just to speak about a portion of your notes where it's asking about the acidity and alkalinity of solutions. So the term neutralization occurs when you mix acids and bases. It's a result of water and a salt. Um, in addition to that, you have some buffers. The chemical systems that maintain your pH release hydrogen ions when the pH rises, and then it'll bind to those ions when the pH drops. So alkalinity or basic substances absorb hydrogen ions. Okay, that was this slide. All right, so let's briefly look at a couple of the buffers that are present within the body. You have the pH of human blood is normal range between 7.35 and 7.45. There are fluctuations of pH outside of this range that can be life-threatening. pH is regulated by buffers, and buffers are solutions of weak acids that have a conjugate base. So it's a mixture to develop a neutralization. A buffer can neutralize small amounts of acids or bases in body fluids. All right, so diving into organic chemistry. Carbon atoms can form single, double, or triple bonds with other carbon atoms. Carbon can form up to 4 bonds, and this allows carbon atoms to form long chains, almost unlimited in length. So at the bottom of the screen, you have several different structures that have carbon associated with it. Here you see carbon is bonded with 4 individual hydrogen atoms. Here we have 2 hydrogen atoms with a triple bond between the carbon. So the number of bonds that are present is represented by the number of lines. Here we had a triple bond, 1 line is a single bond, and when you have 2 lines, it's a double bond. All right, these are some of the functional groups associated with carbon bonding. Hydroxyls are polar and found in all types of organic compounds. Carboxyls are primarily found in fatty acids, amino acids. Um, the the NH2 is found in amino acids. Methyl, which is CH3, is found in also in amino acids. And the phosphates are also found in phosphate lipids and nucleotides. Organic compounds are unique to living systems. We have four organic compounds that are carbohydrates, lipids, also referred to as fats, proteins, and nucleic acids. Organic molecules are, large molecules that are made up of functional groups. The reactions occur within those functional groups. You have monomers, which are subunits of organic compounds, and polymers, which are chains of monomers that make up the organic compound. So water in some of the reactions, we briefly discussed dehydration and hydrolysis. So monomers are joined by the removal of the OH from one of the monomers and removed from the hydrogen ions from other site of the bond formation. So here in this reaction, you have the two monomers being combined, and they release water as they yield a monomer that is linked by a covalent compound. In a hydrolysis reaction, water would be absorbed or produced. So here we have our monomers that are already linked by a covalent bond. And after their reaction, they would be broken into two separate monomers, and the water would be reabsorbed. Some characteristics of organic compounds. Please make notation of these, that they are volatile in nature. They have low melting and boiling points. They are generally insoluble in water, but soluble in organic solvents. They are bad conductors of electricity. They do not furnish ions, and they show molecular reactions. Looking more at carbohydrates, you have simple sugars, those of the monomers that are carbohydrates. You have disaccharides, where there are two sugars or two monosaccharides joined by the dehydration synthesis we just looked at on the previous slide. Polysaccharides are polymers of simple sugars that are linked by dehydration synthesis. Polysaccharides are present more in starch for plant storage and glycogen for human storage of sugar. Carbohydrates provide the body with a ready and easily accessible source of cellular fuel. It is used to make ATP and the cell energy for molecules. So in this slide, we have some images of the different types of monosaccharides and disaccharide structures. Again, carbohydrates are a source of fuel for body cells. They are important parts of ATP, RNA, DNA, glycoproteins, glycolipids, and other molecules found within the body. In later chapters, we will discuss these in more detail. Polysaccharides are important in the storage of energy as glycogen. So lipids, also referred to as fats, are insoluble in water, but they are soluble in other organic solvents like alcohol. There are three types of lipids: triglycerides, phospholipids, and steroids. Triglycerides are three fatty acid chains that are bound by glycerol: fats when solid, oils when liquid. They are nonpolar, so they do not interact with water. They provide the body's most efficient form of stored energy. When they are oxidized, they release a large amount of energy. So fat also provides insulation and protection to our organs. So you can have a saturated or unsaturated bond with your fats. Saturated fats have no double bonds. They are all single bonds that are present. You will find the saturated fats in tropical oils and animal fats. All carbons are saturated with hydrogens. And When it's at room temperature, these fats will be found in a solid state. Unsaturated fats have varying numbers of double bonds. They're very common in plants. All carbons are not fully bonded with the hydrogens. The trains are bent at the double bonded ends and they are liquid at room temperature. So in this slide, you have some information that refers to the differences between bad cholesterol and good cholesterol. I would encourage you as you go through your own presentation to read through this. All right, phospholipids are modified triglycerides. Diglyceride with a phosphorus containing group and two fatty acid chains. This is the main component of the cellular membrane because of its polar head and nonpolar tail. So the polar head is the portion that is water loving and it will readily react with water, whereas the nonpolar tail is water phobic or water fearing and non-soluble with interaction with water. Again, we will look at some of these um, different types of carbohydrates in later chapters. So steroids are another type of lipid. They're different from fat in structure and function. Carbon skeleton is bent to form four fused rings. Cholesterol is the base steroid, and from which your body produces. There are other steroids. Some examples are sex hormones such as testosterone and estrogen. Cholesterol is the most important steroid in the body. It's needed to produce cell membranes, synthesize our vitamin D, and then the steroid hormones, as we just identified as testosterone and estrogen. And it is also present in your biosalts. Protein is the basic structure material for the body. Amino acids are the building blocks of those proteins, and there are 20 different types of amino acids. They are the base structures of an amine group and an acid group. The R group is designated with the specific types of amino acids. When amino acids are binding, they are connected to each other by dehydration synthesis, and they're usually present within peptide bonds. There are different levels of protein structures. You have primary, secondary, tertiary, and quaternary. The first sequence of amino acids, uh, the second is a hydrogen bond of the peptide, peptide backbone that causes amino acids to fold into a repeating pattern. That secondary, tertiary portion is very prominent in your DNA. Tertiary protein structures three-dimensional folding patterns of proteins due to side chain interactions. You'll see some examples of that with your RNA chains. And then the last protein structures, protein consistent of more than one amino acid and chain. Enzymes are globular proteins that act as biological catalysts. The enzyme substance regulates and accelerates the rate of reactions, but are not themselves changed or used up. Catalysts are types of enzymes that that can be purely protein or can be made up of two parts. And the substrate on which an enzyme acts on. So enzyme action: for every reaction, there is an activation energy that must make it go. Enzymes decrease the amount of activation energy that is needed. With our nucleic acids, the nucleic acid includes DNA and RNA. Nucleic acids are macromolecules that are formed when long chains of molecules called nucleotides join together. The order of DNA and RNA is important. and work as codes for instruction. These instructions tell the cell how to grow, how to reproduce, how to respond to environmental changes, and how to make proteins. Here we have an example of a single DNA structure. You have your monomer, which is the base subunit, the nucleotide, and the phosphate. So this base is our nucleotide. The monomer is our phosphate group. I'm sorry, it's our 5-carbon sugar deoxyribose, and then our base. I'm saying it wrong. This is the base, which is the nitrogen base, which is one of your nucleotides associated with the DNA. This is the deoxyribose sugar, and then this is the phosphate. With DNA, your groups of nucleotides are either 2-ring bases or single-ring bases. So adenine and guanine are the purines, which have 2 rings, and the pyrimidines have single rings, and you will have cytosine and thymine. If you are looking at an RNA strand, then uracil will replace thymine, and we'll look into more detail on that in our live discussions. When you look at DNA versus RNA, typically the structures are very similar. However, DNA is double-stranded and RNA is single-stranded. At the bottom of this slide, you see the nucleotides that are present in each. So both DNA and RNA are identical until you reach thymine. Thymine is not present in RNA, and uracil is not not present in DNA. The shapes of the nucleotides, again, DNA has a double helix shape and RNA has a single helix shape. ATP is adenosine triphosphate. This is the energy molecule that is used by cells that usually come from glucose. The energy exists in the bond between the oxygen and the phosphorus. There's a terminal phosphate that is transferred to other molecules, phosphorylating them and then energizing them. All right, so the only thing that we were not able to discuss here were the construction of RNA and DNA strands. I will provide a supplemental video in case we are not able to discuss this process in our live sessions. Thank you and have a great day.